tips in space. What's nice about the mouse lung is that it's quite stereotyped. So you take two lungs from different mouse, and they look very similar to each other. So they can identify a whole hierarchy of tips, and they put names to those. When I say they, biologists, they take the tissue and can classify each of the branches, and they can also find different ways of branching. So some of them include lateral branching. So you have the duct of the tissue, and even from the duct, there's new tips coming out. There's tip branching events. Those are the ones I'm going to focus today. So you have tip branching, that is a domain branching, where one duct forms a whole lot of new tips on a plane. There's also some correlated modes of branching, where a branching event occurs in a plane, but then the next one is correlated to the previous one and occurs in a perpendicular plane. And there's been studies relating that to release of mechanical stresses in the tissue. So when a tip divides, it chooses the way of division in a way that it releases stress. And then the next one must be in the opposite direction so that it continues releasing stresses, okay? But that's maybe not controversial, but there's discussion on what is really driving these different modes of branching in the mouse lung. But something that we're all agree on is that this process of branching morphogenesis is a multi-scale process. So it involves everything from the cellular dynamics in the tips. So here in this talk, the tips will be our main object of study. Tips hold all the cells that are dividing in the tissue. So this is an epithelium that is dividing, and the active tips will be in the tip. And as they divide, they're gonna crowd the space in the tip, and some cells are gonna be pushed back to the ducts, and that's gonna cause the elongation of one of these ducts. At the same time, because you have cells and you have mechanical forces between them, there's some tissue tension. Because they're forming a lumen, so they're forming a connected ductal structure, there's luminal pressure. Because they are growing within a matrix that is a mesenchymal tissue, then there's interactions with the extracellular matrix components, and there's also signaling, so there's communication between the epithelium and the mesenchyme. So all of this is just to show how complex this system is in general. Any system that involves branching morphogenesis is a complex system. But as Elizabeth was saying early on, we're physicists. We want to dissect this into hopefully a few set of rules and see if that's enough. And then tell the biologists, well, this has nothing to do with global guidance. This is all regulated at a single tip level, for example. So we can go and understand what are the rules and the cues, right? The molecular cues that are driving these processes. So what are the rules that we can identify from a tissue dynamic like this? Well, the first thing is elongation. So we need these tips that are going to be our objects of study. We need our tips to grow, right? And if you think of our active particles, some people were talking about active particles early on. An active particle is a particle that has some persistent motion, and it's going to move in one direction. In this case, we have something similar, but at the same time, the tip is marking its way. It's leaving a trace, so it's leaving memory of the places it has visited, right? So it's going to be our elongation process, and it's driven by the division of cells in the tip. At the same time, we need branching, and this is tip branching, so we're going to study tissues where tip branching is the main mode of branching. And in that case, what happens in the biology is that the tips accumulate elements from the extracellular matrix, then cells from the mesenchyme migrate into that cleft, and they force the division of the tip into two new ones. In terms of physics, we can think of that as just two particles being born from a single one, right? And of course, because in these tissues we don't have reconnections, so if you think of the lung doesn't have loops, right? So we need a mechanism that inhibits reconnections. So what you see there, I can't really see there it is. So whenever a tip grows into existing duct or other tips, it's going to be inhibited, and we can discuss how that inhibition occurs. It could be mechanical, it could be molecular signaling, but what happens in the tip is that cells exit cell cycle collectively. So they're trying to grow, they encounter some signal provided by the rest of the tissue, and they just stop dividing. Yeah. So we have 3 rules here. And if I give this 3 rules to any of you, you could say, well, I can simulate that, right? I can put that in a computer in a very simple 2D lattice and see what happens. So let's see what happens, right? Yeah, exactly. So this is another video. I hope it runs. Yeah, thank you. This is a hexagonal lattice. It's a very simple simulation, but it really highlights what the rules are going for, right? So what's happening here is that we started with a single active cell, agent or particle. That particle is proliferative. It means that it wants to explore the space. It moves persistently, as persistent as you can in a triangular lattice. So this means that it's not going to go backwards. Once in a while, stochastically, it's going to branch. It's going to divide into 2, form 2 new ones. And when any tip encounters the dactyl structure, it stops, right? You can see that the only stationary state here in a finite system is a dead system, right? We can only have dactyl structure at the very end. All active tips have died out, right? So we don't have any more of that. What we're left with here, some people call it optimal. That's what's optimal in this sense. It depends on the function of the tissue. But what's clear is that we are not filling the space, right? We're leaving behind holes, and the sizes of those holes depend on the fractal dimension of these objects. There's a whole literature on this. And what people have studied, though, is that this process is interesting from a reaction diffusion perspective. And already in the 80s, when the boom of statistical field theory came in, already Cardy, Grasberger, Janssen were discussing this type of models because they look like epidemic models. So if you think of the lattice as agents that can be infected, then the particles that I draw there as my dactyl structure is a virus that is propagating through space, and when it tries to infect an already dead agent, then it can't do it, and you end up with this type of dynamics, right? And what we've learned from the statistical field theory side of things is that the upper critical dimension of this object is 6. This means that correlations are relevant all the way up to 6 dimensions. We know what the critical, the universality class is for this object that is propagating. So if you're more into the StatMech side of things, the universality class of the propagating cluster is dynamic isotropic percolation, and what you're left at the end is just a percolating cluster. Yeah, standard percolation. Yeah, so we know these things from the physical side of things, but we want to know if these rules apply to any tissue at all, and we know it does. So from previous work, we know that these models of growth apply to tissues. I put simple there. Nothing in biology is simple, of course, not even bacteria. So you can imagine what simple means. It just means that it's something that we can model with a simple model, right? And I'm going to talk very briefly about the mouse mammary gland. Why I want to introduce this? It's because this is a 2D tissue. So I want to go from 2D to 3D. Yeah. So in a 2D tissue like the mouse mammary gland, this tissue develops on a fat pad. So it's a thin layer of fat where the dactyl network develops. And if you do experiments on this, and this is work from Schill and Haneso from back to 2017, what you see is that the tissue, sorry, can you see there? The network is the gray lines. They look like they're overlapping, but in reality, they overlap very little. Yeah, so they do terminate as they encounter each other. And this means that when the network develops and it encounters edges, it just stops proliferating. And this means that all proliferation, which is here marked by EDU, that's a proliferation marker, it marks cells that are in active cell cycle, they all localize in the propagating front. And that's exactly what we saw in the model early on. And if you run the model on a domain like this, you're going to see that what's left behind is a dead network, and then on the front you have the active tips proliferating. So qualitatively, it reproduces the self-organization of active tips on the propagating front, right? We want more than that, of course. We want to see if topologically the networks are equivalent, and if they, if quantitatively we can use these models to describe the dynamics of the tissue. You can see here also that the model here is continuous in space. We lost the lattice. Of course, we want to analyze something that is continuous in space. And here we, again, if we take our set of reactions, this is active tips that branch into two, and then we have an elongation rate where an active tip deposits, that's a memory, right? It's depositing inactive particles. And at the same time, when an active particle meets an inactive particle, it's going to get inhibited, and it's going to produce an inactive particle as well. And what you can get from that, if you build a set of PDs that describe the evolution of A and I, so the concentrations here, at the mean field level, what you see is that this model reproduces the propagation of a pulse of active particles, and is leaving behind a homogeneous concentration of inactive particles. And when the system is fully explored, then you end up with only a uniform concentration of inactive particles. Yeah. I'm not going to go into many of the details because it's not the aim of this talk, but I want to just convince you that this model does work. So if you, with no fitting parameters, basically here, you can get and reproduce the distribution of branch lengths that is stationary. You can reproduce some parts of the topology of the tree, so how many tips terminate at a given level. You can, and because this is a stochastic model, you immediately get an exponential distribution of duct lengths, right? Because tips propagate ballistically, and if they branch stochastically, then the lengths are exponentially distributed. So overall, this model works, and that gives us a bit of hope to be able to explain more complex tissues, and that's what I want to ask now. Yeah? So what's more complex than 2D? 3D. But it's not like we can just grow a mammary gland in 3D during development in the mouse. It doesn't work like that. So what we did is that we explored a tissue that grows in 3D, and that's the mouse salivary gland. So the salivary gland is interesting for many biological reasons and even medical reasons. It's an interesting case study. For us, it's good because it's a tissue that also develops through the branching of active tips. Yeah. But at the same time, that's a video as well. If you see its configuration in space, and maybe it runs now. Yeah, it runs now. Yeah. So if you see its spatial configuration, it's a 3D, but it's quite flat. So it's wide and not too thick, right? It's like a pancake. Yeah. So it's a tissue that is sufficiently complicated so that we have to improve the theory, but it's sufficiently simple that we can analyze it reasonably well in a decent amount of time. Yeah. So we can segment this in. What we did was to segment the salivary gland at different stages of development, and you can see there, I just put 3 examples. This whole structure goes from day 14.5 in embryonic development of the mouse, and that's 18.5, so this happens in a matter of 4 days. These are not the same glands, so we cannot do that. We cannot trace them for that long in vivo. We can take the tissue out and analyze it ex vivo, but this was, so we have to take the salivary gland out of the mouse and then image it. So these are different samples. But you can see how it goes from just 100 ducts to almost 4,000 ducts. Yeah, so it really expands overall. So the question is, if we do some statistics of this, does it follow the rules that we need or we implemented in the simplest branching annihilating random walk dynamic? And there's already, there's some things that gives us hope. One thing is that there's heterogeneity in the subtrees. So if we look at the subtrees up there, so that's what's the sizes of the trees that grow after a given generation. We see that that's heterogeneous, that's compatible with a stochastic model of growth. But then if we look at other statistics, for example, we look at proliferation in bulk our model, a previous model, tells us that in bulk there should be no proliferation, right? Because that's just dead tissue in the sense that it's not proliferating anymore. But if we measure that experimentally, we see that proliferation occurs everywhere. Even these are submerged tips, and these are tips at the periphery, and the difference in proliferation is very little, but it's lower in bulk. At the same time, all our topological quantities, like how many tips are inactive at a given generation, those keep changing with age. So they're not fixed even in the bulk region. Yeah, that should happen in a tissue that is leaving behind an inactive ductal structure. So what I did here was to go and see what was happening with the tissue. And the actual tissue, if you look at its size as a function of time, you see that the tissue is growing exponentially. So now we have proliferation everywhere and an exponential growth of the tissue. If we have proliferation everywhere, that means that the tissue is inflating over time. And the fact that it's expanding exponentially means that the tissue is, every 2 points in the tissue are separating exponentially from each other. It's like an inflating universe, right? It's the idea that every point in space is getting further away faster in time, yeah? So the question that comes now is, how do we implement this new set of rules into our branching annihilating random walk? And that's not too complicated if you think that what happens in our original model is elongation, bifurcation, and inhibition. Whenever you have inflation, if you have inflation of a tissue that is exponential, then submerged tips can acquire space, and that means that they will continue their growth process and branching dynamics. So even submerged tissue can continue contributing to the branching process. This is not entirely true if the tissue grows linearly. That's the ant on a rubber band problem, right? Because the tissue grows exponentially, you're going to have a significant amount of contribution to the network coming from bulk, yeah? And that explains many things. Well, we can run the numerics now. We can see how the tissue develops. I'm starting here from an initial condition that is experimental, but then I'm simulating the growth of the remaining 4,000 ducts. And what we see here is that a simple model like this can reproduce. I put here just a few topological measures, but also morphological measures of the ductal structure. So now we can reproduce what's the distribution of subtree sizes, what's the probability of seeing terminal ducts at a given level, just by incorporating growth into this process, so inflation into this dynamics. But now we want to know if we can do something a bit more analytical with this, right? So we could, I'm not sure what time is it, but I think I do have time. I do have time, right? Yes. So, just before going to the mean field, there's something that our model predicts, and that is that proliferation and branching should continue occurring in the bulk of the tissue. Slower than in the periphery, but should continue, right? Because in bulk, tips are being arrested by the neighboring duct, and only when stresses are released, then proliferation can continue. So what we did, this was Lemonia, of course, my experimental collaborator. What she did was an explant. So this is amazing because they take the organ out of the body of the mouse, and then they culture the organ ex vivo. So they let it grow for, in this case, was 40 hours, I think. Yeah, 40 hours, and image it every 1 hour and see what the tissue does. So this is quite impressive because here you can also see this sort of inflation of the tissue as it develops. And what we did there was to quantify how little trees of the ductal structure behave when they are near the periphery. So this is a bit, it's a bit techy here, but just believe me when I say that black dots are tips that are submerged, blue dots are tips that are close to the periphery, and this is the tree that is reconstructed from those images. Yeah, and what we see is that after 40 hours of growth, the tips that were submerged produce only a handful of new ducts, Actually, they produced 1, 2, 3, 4 new ducts, while tips that were in the periphery, those are the blue ones, they produced very large structures of ductal structures, right? So this somehow confirms the idea that submerged tips can continue proliferating, but at a lower rate than those at the periphery. Yeah. So let's see what we can do now with our model. Let's see. So we have active tips. Those are A that form 2 tips. So that's the branching event. So we have the deposition of ducts. This is known. This is exactly the equations that we had before. We have all these inhibition equations. Here I incorporate interactions between any 2 types of cells. So active and inactive, active and active, and this new type S. S will be cells that with a given rate can recover the active state. Yeah, so this is the difference from the previous theory. So we have now our branching, duct deposition, tip, sorry, I'm colorblind, so it's really hard to see the red one. So then we have tip reactivation as well, and that's going to happen with a rate that is proportional to the inflation rate of the tissue. So what we can do is that we can build our set of PDEs from these equations, but here the only thing we've included is local effects of the inflation. So we have altered our rates. We have now 3 equations for the concentration of active, arrested, if you want, and inactive tips. And we have how the reactions are going to be changed by the fact that the tissue is inflating. But now we want to incorporate the inflating tissue, right? Inflation means transport of particles, and that's easy to incorporate if you think of the inflation as a flow, right? So a cell that is here or an active tip that is here is going to be transported away by the flow caused by inflation, right? And that incorporates 2 terms because we have a product of the flow times the concentration. One is that actual drift, and the other one is the decay in concentration. So it's a dilution term. It's a decay in concentration because there's inflation. So if we don't have enough branching events, the concentration will go down to 0 if we just let the system inflate. Yeah, it's the idea of in the universe as well, right? The cool down and everything. If we inflate too much and we don't have energy to fill the space, then we just reach a minimum energy state. So the good thing about inflation or this expansion of the system is that it introduces new stationary states in the system. So previously I said the only stationary state is of course the dead system, but if I let the system evolve in an infinite system, then we only have a pulse of active tips at the periphery, but all the bulk is dead. But here, because we have inflation, even bulk can continue its activity. So here I'm just going to solve the previous PDEs, and you're going to see that as active tips have this peak at the periphery, but what is being left behind here is just a uniform concentration of active tips, but also inactive tips and arrested tips. So there's an active bulk as the tissue develops. Seeing this peak at the periphery is interesting. And here we're in the co-moving frame of reference. The system is inflating, but that's why you don't see X changing with time, because we've rescaled our variables. But what it's nice is that if we go back to the experiments and quantify, let's say the number of the level of proliferation of the tissue as a function from the distance to the edge, right? So in the previous model, we would have seen a pulse here, but what we see now is an increased concentration at the periphery, which then decays into something that looks approximately constant in bulk. Yeah, so bulk is down here, periphery is down here. So that's consistent with the predictions of the model. So now we have both a very simple extension of our model is able to capture both the topological and morphological features of the tissue, which is quite remarkable thinking back on the complexity of what's going on actually in the tissue. So I want to finish with a few remarks. These are just some minimal concluding remarks. I hope you take something home. It doesn't have to be this, but something. I'm working on other things now that have to do with the actual organization of these tips, and for that we need a model that can explain why the tips behave in the way they do, and how a tip can survive multiple rounds of branching. In general, you think of this branching event as a very it's a strong perturbation to the dynamics of the tip. So how are cells not exiting cell cycle in that context, or being altered significantly in a branching event, and how they can continue doing this serially over time is after, as there was a continuous transition between a more fluidized behavior of cells in the tips and then a more solid-like behavior in the tips, and that repeats over time. We know that from images of the tips. Yeah, so that's something I'm working at the moment. I just want to thank. Most of this work was done by, the experimental work was done by Lemonia. This was done when I was a postdoc, still in Ben's group, but I continue working on this independently now. Part of the field theory has been done with guys at Imperial, and a shout out to my students, some of which will start working on this kind of problems. Finally, postdoc opportunities. If anyone is finishing the PhDs around these dates, you can go to Chile. It's a nice place. Spend a year over there. I don't know, do something fun. And you can work in experimental, theoretical things, numerical things, tissues, bacteria, I don't know, many things. And the other thing, it's not here. Yeah, but there's a school. We're organizing a school for last year students in July. Yeah, so if anyone wants to visit Chile for the first time and you're a final year undergraduate or master student, go to Chile. I'm gonna be waiting for you. Yeah. Thank you.
Thank you very much, Ignacio. So we have time for questions.
Thank you for your talk. I was wondering what causes a tip be inactive or active, and the tip growth stop in.
Yeah, there's. It's probably a combination of mechanical. In the numerics, you mean, or in the actual biology? Okay, in the numerics, it's very simple. In the model, when you implement it on the lattice or in continuous space, you do something that is deterministic. So whenever a tip is closer than a given radius to the network, then you just make it stop with probability 1. Yeah. But when you do the reaction diffusion type of model and the statistical field theory side of things, then you need rates. Yeah. So you're gonna stop with a given probability that is, of course, proportional to the rate that you put in there. So the two things are not exactly the same, only in the limit when the rate of annihilation is infinite. Then you recover the biology. So it's a tricky limit to take in the theory. In the biology, it's probably a combination of mechanical and molecular cues. Yeah. So in the lung, for example, I think FGF is one of it's a growth factor that regulates growth of these cells, so it allows cells to continue dividing. But when you're in a crowded environment, the concentration is lowered, so cells detect that and they exit cell cycle. They stop proliferating because they don't have the signals from the mesenchyme. Yeah, so it's tightly regulated there. It's like a quorum sensing mechanism. They sense that the environment is too dense with epithelial tissue, and they just stop dividing. Yeah. Yeah.
Hi, thanks for the talk. Seems to be a quite universal model that you made, that the tips keep splitting and seems to be working for multiple organs. Have you tried to look into the fractal dimension or anything to try to see if there is any universality in the growth of different organs or tissues of different animals? Or
Yeah, so people have looked at the fractal dimension of lungs, vascular networks, and all sorts of things. But that's usually done in adult tissues. And there's striking differences between, for example, I'm talking about lung now, the developing lung and the adult lung. There's a whole remodeling process that takes place because of the function the function of the lung when you're born, right? There's this Murray law type of behavior where ducts have to acquire certain widths and lengths so that dissipation is minimized, and that is true in the adult lung, for example. And their fractal dimension of the adult lung is like 2.9 something. It's very close to 3, but it's not, of course. And people have done that. But that should be a result of the dynamic. It shouldn't be something that I put into the model. And if you look at these models, because they belong to the universality class of percolation, for instance, you already know what is going to be the fractal dimension of the cluster that is being formed. So we know those things. The statistical field theory breaks down at dimensions 2, so the predicted exponents. If you work at the critical point, but it works in dimensions 3 and above. So many of the observables break down at dimensions 2 because of how they work. But we know those things. Yeah, it's interesting.
Do you know what happens to this exponential growth regime when it encounters a boundary, some other tissue it can't grow past? Do you expect a difference in the dynamics as a function of time because of that?
Yeah, the stopping process is not so trivial. Of course, in the numerics, it's trivial, right? Just put a boundary and see what happens, how quickly it decays, and it decays exponentially, the tissue to the dead state. But in the biology, it's difficult because the tissue enters a different state, a different stage of growth, where these tips enter a sort of budding phase. So now every tip divides into multiple acini. The acini are this sort of sacs that produce saliva, and then they're being pushed, and the saliva, they compress, and the saliva is being pushed out. Yeah, but that's it's like a grape. It divides into multiple of those things. So there's probably some sort of signal. I'm not sure though. What's the actual dynamics when it's stopping? Yeah. The same happens in the lungs. You have a budding phase where you form all the alveoli.
Yeah, just in your reaction diffusion equations, you put the velocities there, so you just take in the constant velocity.
Yeah.
Okay, so it's just an external parameter. But you can imagine that it's probably going to slow down maybe as it grows also, so the velocity.
Yeah, that's what I was saying. So implementing a slowdown in the model is trivial. But what's really happening in the biology, that's more complicated. Yeah, we can put a slowdown or boundaries or whatever we want, and that's gonna tell us how the system equilibrates later on. But that's not exactly what's going on in the biology. Yeah. We don't have data of that actually. We never analyzed that later, those later stages.
More questions? So if not, let's thank Ignacio again.
Thank you.
Okay, the last speaker cannot make it today, so we'll go to the coffee break now. Let me say that there are some seminar rooms that you can use for discussion, for instance, if you are interested. And they are very close to where the coffee break is, so feel free to use the space to discuss science and just things like that. Thank you very much. Okay.